Good morning, everybody. I'm very, very glad to have you and to welcome in Budapest. Believe or not believe that the story that we are here today, it started more than 30 years ago. It means that in the early 70s, Albert Szent-Györgyi, one of the Hungarian Nobel laureates, said that we should go to the submolecular level. He wrote in an article, the title was Living State and Cancer, that the present day biology is a molecular biology which searches for answers mainly in the molecular dimension. Our body is built of molecules, so its reactions have to be molecular reactions, but molecules are built of atoms, and atoms are built of nuclei and electrons. So there is another dimension below the molecules which has been disregarded by biology. And later, he wrote that I could never believe that the wonderful subtlety of biological reactions should be brought about by clumsy, relatively unreactive macromolecules without the concurrence of much smaller and more mobile units which could hardly be anything else than electrons. So Albert Szent-Györgyi suggested almost 40 years ago that the electrons have some malfunctions, and the stream and then the flow of electrons has some problems, and that can cause the cancer. At that time, I was pretty young, and in 1976, 21st of May, I made a note, and this is a note that I wrote 34 years ago. Those who are not Hungarian, I just mentioned that I cite Szent-Györgyi, who said that the electron has some central role in the living organism, and at that time I saw that not the negatively charged electron but the positively charged proton has a key role in the cancer development. And I kept it in my mind for 4 years. 4 years later I was a student at the biological faculty, and then the other idea came to my mind that the deuterium also a hydrogen, and then I don't know why, but I was almost sure that somehow the hydrogen-deuterium isotope pair can be the key in the regulation of cell division. And 10 years later, I started to work at the National Institute of Oncology, and the reason I went there because I saw that my idea, whether it's a good idea or there is some scientific background, and we can prove it that it's a good idea. So research started 20 years ago, and now 20 years later we are here. And we are very, very happy that so many people come together. And the real point is that several scientists are here who has made experiments with using depleted water. And I hope that by the end of the day, everybody will be convinced that this is a good direction, and we have to make more experiments on that here. So when I look at the attended scientists in this room, I think these are the main characters of a new chapter of science, and those who recognized in the past that the importance of nature occurring deuterium and carried out several experiments with this discovery. I think these are the key persons knowing that story. I think that symposium, not only a symposium, that symposium is the #1 symposium with deuterium depleted water. And those who are here, I hope, will be proud for that they took part in the first international symposium on deuterium depletion. Today, there will be 15 presentations presented by 12 scientists. There are 31 co-authors of these presentations, and people out of Hungary come from the United States, from Romania, from Russia, and from Iran. And they will also present beside the Hungarian scientists their results. So I wish you a prosperous meeting, a successful meeting, and I'm glad to open the first international symposium on deuterium depletion. We have a very busy day today, so I will ask all the chairmen to be very strict, and I ask everybody to keep the time and save some time for the discussion as well. We have a small bell, and I ask the chairmen to ring the bell, everybody in time, to be sure that we can keep our program. So the first section will be chaired by Professor Janscha and Professor Signac, and I ask them to chair the first section of the conference.
Welcome to this meeting, and we will start with the first session. Do you don't hear? You don't? Well, how to work?
It already works.
It already works, sorry. Okay. Well, the first session will start with the first speaker, with Professor Somjai, and he will talk about naturally occurring deuterium is essential for the normal growth rate of cells. Please.
Thank you. I should find the... There is?
You are the first speaker, sorry.
I know, sorry.
You are not the one.
On my computer I put here.
F5.
F5, okay. Még egyszer?
Slideshow. That's slide.
Okay. Ladies and gentlemen, the title of this presentation, Nature Occurring Deuterium, is essential for normal growth rate of cells. The next presentation will talk about in detail the hydrogen deuterium. Now I would like to flash up a couple of slides. So we know that the deuterium is heavy hydrogen. We know that due to the 100% mass difference, the hydrogen behaves different in chemical reaction. And we also know that in a normal surface rate water, the deconcentration is about 150 ppm, which is equivalent with 16.8 millimole per liter. And the real question, what I addressed long ago, we can consider that the deuterium concentration in a living organism is 12-14 millimole per liter. And if we compare it with the calcium concentration or to the magnesium concentration, we can say that the deuterium concentration is pretty relevant. And the question is whether this deuterium has any role in living organisms or not. To answer for that question, we have one way. If we remove the deuterium or we reduce the deuterium concentration, and we see whether it has any effect on biological systems. So the very simplest and the most simplest experiment, if we use deuterium depleted water to prepare media and to see whether it can modify the cell growth in vitro. That was the very first experiment when we used L929 cell lines, and this is a normal deuterium level, this is a 30 ppm, and we could see that deuterium depleted water or media inhibited the growth of cell, and we also could see that slightly increasing the deconcentration that could stimulate the cell multiplication. And other experiments were carried out concentration that could stimulate the cell multiplication. And other experiments were carried out by Gábor Laskai. In that experiment, we used another cell line A4, and here we can see when the cells were transferred from normal water to deuterium-depleted water, I mean media. In the deuterium-depleted media, the cell growth stopped for a while, and after 15 hours, the cell growth started again. Another experiment with the HT29, this is a colon tumor cell line. We also could see the dose effect. This is a normal deuterium concentration, and the lower was the concentration, the bigger was the inhibitory effect on cell growth. Then we went to the United States and asked the Oncotech Incorporation, which is testing several compounds for big pharma, whether it has any antiproliferative effect. They tested three different cell lines, PC3, prostate, breast, and melanoma cell lines. And they also could prove that deuterium depletion inhibited the cell growth of the cell growth. All these in vitro experiments, we reduced the deuterium concentration in one step. Later we asked them we asked the scientists to reduce not in one step the deuterium, but in 5 steps. And the next figure shows that when it was reduced twice within 24 hours, 3 times, 4 times, 5 times within 48 hours, or 5 times within 72 hours. More times the deconcentration was decreased, the bigger was the inhibitory effect. It is with one cell line, and with the other cell line, we could see the same effect. So that could be something similar what we can experience when people or animals consume deuterium-depleted water, so that there is a gradual decrease in deuterium concentration. We tested a not humorous cell line, which was a human myometrial cell line. Here we also could see some effect, but it never was so big than comparing to the tumorous cell lines. In the very first animal experiment, we used a breast cancer tumor. We transplanted to mice. At the beginning of the experiment, we had 17 mice in the treated group and 11 in the control group. And 87 days later, in the treated group, which were consuming deuterium-depleted water, we had 11 mice without any tumor. Here, all the animals has perished, except one, which also still has the tumor. In that case, the treatment started right after the day that the tumor had been transplanted into the mice. In another experiment, we used mice, 22 mice in the control group, 22 mice in the treated group, and the prostate cancer was transplanted into the mice. In the first 32 days, both groups consumed normal water. At that time, the average tumor size was the same in the two groups. At that time, we changed the normal water for deuterium-depleted water, and you could see that in the control group, the tumor growth was very intensive, but it was slowed down, and it was much smaller, the growth rate in the treated group. At other experiments, we used again the PCC prostate cancer. The tumor was transplanted. In the first 18 days, both groups consumed normal water. Then the treated group consumed deuterium-depleted water for 12 days. After that, we killed the animals and we checked the cells being in mitosis and mitosis in the two groups. We found that 3.6% were in mitosis and only one in apoptosis in the control group. And the average tumor size was 3.7 cube centimeter. In the other group, that ratio was just the opposite. So the deuterium depletion triggered apoptosis, and the tumor value was very much smaller, and the shape was a ball shape. Here, suggesting that somehow the deuterium depletion can trigger the apoptosis. Gábor Laskay made another experiment in vitro with that similar system. This is an interleukin-dependent cell lines, which means when we withdraw the interleukin, that triggers the apoptosis. And here we checked that process in normal water and deuterium-depleted water, and the figure shows that somehow the deuterium depletion even triggered the apoptosis, suggesting again that somehow this deuterium could be very important for the cell. The question is, how can we explain our results? If we go back a couple of decades, we can say that it is widely accepted that before the cells start to multiply, there is the activation of sodium-hydrogen transport system, which causes a pH shift, and that shifts somehow a message for the cell to multiply. There was a paper published in the Nature that article says that they isolated the hydrogen ATPase gene from yeast, and they used that gene to transform animal cell line, and they could prove that the cell expressed the gene, and the ATPase were sitting in the membrane and throwing out the hydrogen, and they proved that the pH shift has occurred. But everybody was surprised that that tumor showed tumorous phenotype in vitro. So nobody could explain how is it possible that the ATPase gene behaves as an oncogene. The real answer we can get if we can suppose that the life can make difference between the hydrogen and the deuterium, and we check for discrimination of isotopes, we can find several papers saying that the lives can make difference between the two hydrogen isotopes. For example, the large hydrogen isotope fractionation between the liquids and gross water between what we observe in most organisms, or hydrogen isotope discrimination in higher plants, depending on what biochemical way is used to fix the carbon dioxide, or deuterium discrimination between a phosphor hydrogen, phosphor deuterium exchange of phosphorus acid. So it is clear for today that the life is so sophisticated that can differentiate between the hydrogen and deuterium. But it can be very important for us. There is a paper which says that the hydrogen ATPase do not accept deuterium as a substrate. So it means that these enzymes are so sophisticated. This is a final signal for the cell, changing the pH ratio. And when we are using deuterium depleted water, even that system is activated, but the cells are not able to lift up the pH ratio to the threshold, or it takes much longer time. And this is the reason that we can finally disturb the whole metabolism and no signal and no cell division, and finally we can trigger the apoptosis. There was another experiment which partially can prove that, and Gábor Laskay will show another one. In that experiment, we made an acid load. So we brought down the pH in the cell to about 3, and then we checked the amyloid sensitive sodium hydrogen antiport, how can it work and how can recover the pH in the cell. And as you see, the curve is different depending on the deconcentration. So we can suggest that somehow, for example, the sodium hydrogen antiport is sensitive for the deconcentration, and that can modify the activity of that transport system. I have shown you that in vitro growth, the lower was the deconcentration, the bigger was the inhibitory effect. But the real question was whether we can modify the expression of COX-2 gene. It is well known that the COX-2 gene has a key role in tumor development. It is responsible for enzyme which synthesize prostaglandin. And what we could see here, we could see a clear correlation between the deuterium concentration and the expression of COX-2 gene. And at the same time, we found correlation with the prostaglandin concentration and the deconcentration. The lower was the deconcentration, the lower was the lower was the deuterium concentration, the lower was the concentrated prostaglandin. And the question was whether this prostaglandin has any role in this inhibitory effect. So we made another experiment that we add back the prostaglandin from outside, and we checked whether we can diminish the inhibitory effect of deuterium, and we could do that, because as we increased the prostaglandin concentration, simply disappeared the inhibitory effect of deuterium depleted water. So that could be one example that the deuterium depleted water can intervene into that signal system and can modify the enzymes and gene expression which are closely related to cell growth. In another experiment, we just checked the MAP kinase, which is labeled within 2 minutes when the cell got a signal, and you could see it didn't happen when we use deuterium depleted water, suggesting again we can modify this system. Later we went on and we chose the DNA chip containing all the 30,000 human genes, and this is the results what we got. It was what we found at the first sight, that a couple of hundred gene expression were changed in deuterium depleted water. So that answer was so complex that we decided not to go and not to continue in that direction. We chose another DNA chip with only 500 different genes. These were kinases. And repeating several times and choosing the real good responding genes, we finally could say that 7 genes responded extremely well to deuterium depletion. There was an up regulation and there was a down regulation. Combining all these results and all the results of the experiments, we could say that there is an extremely complex response to deuterium depletion, and I think there are a lot of work to do and to discover all the background of this process. So the conclusion, we say that the naturally occurring deuterium may have a key role in cell cycle regulation. The changing DH ratio can be the signal to introduce to the induction of numerous cell process. The changing DH ratio can simultaneously modify the expression of genes and the activity of certain enzymes. We say that the results show that it had unknown submolecular regulatory systems may exist in the cell, and this deuterium hydrogen submolecular regulatory system can be a new target for anti-cancer drug development. Before I finish my talk, I would like to ask you to think about, if we take only one animal cells, we can see how complex its structure. We know that within one human cell, there is a DNA with a 1.8 meter length. It contains 3,200,000,000 base pairs, and all this DNA, for example, is packaged in a small cell which size is 40 micrometer. We have discovered the metabolic pathways. It's extremely complex. The question is how the life can organize that extremely complex system. And I say that should be the submolecular regulatory system. And I think if we can proceed, we can make a big leap. So used to we use a computer and with a huge piece of equipment, and it could make only a couple of calculations, and that chip can do maybe 1,000 times, million times more than these big, very old computers. So I say that the life can organize that very, very sophisticated system, because on a submolecular regulatory system, they can simultaneously organize the gene expression and proteins. And that way, somehow, they can harmonize all the processes in the cell. That's all. Thank you for your attention.
Thank you for your presentation. The paper is open now for discussions. Any questions or comments, please?